All right, welcome to your video lecture, Module 1.3a. We're going to be talking about cells of the nervous system today. In order to begin, we are going to talk about the difference between glial cells and neurons. And so kind of the easiest way to think about the difference between them is if we look at these pictures that I have on the screen here, kind of the difference between the role of the staff members in the school versus the role of a student in the school. So students, your job is to actively learn, think, and you're kind of going to be passing information to each other, meaning you're going to obtain information and then you're going to pass information along in like when you take tests or when you turn in assignments. Versus teachers, janitors, cafeteria staff, tech support, we don't necessarily take any tests. We don't pass information to pass classes. We don't have to do those types of things, but rather instead our job is to make sure things run smoothly in the school community so that you students are able to be successful and get that information and send that information that you learn. And so our job in the community, the school community, is to make sure that you have all the things that you need to be able to do your job. And so that's kind of the difference between glial cells and neurons. So neurons are electrical communication that happens in your body where it takes in and sends information out, sending different messages throughout your body to help you with being able to move muscles, to being able to experience feelings, to be able to allow you to learn. So these neurons communicate with one another, similar to students kind of communicate by getting information and communicating that back to their teachers. In contrast to that, glial cells, their roles are solely to be kind of the support cells of the nervous system. Glial cells don't fire action potentials. They aren't like a neuron where they take in and receive messages. Rather, they are in charge of kind of providing a nutrient and oxygen to the neurons, guiding the neurons' growth, helping to make sure to take care of the environment so that the neurons are able to function. So our focus for today is going to be on the neurons and what are the parts and functions of a neuron? How does a neuron fire? What are those chemical messengers of a neuron? So that's what our focus is today. So jumping right in, we're going to look at the parts and functions of a neuron. So just looking at the structures themselves is where we're going to start. So this is a nice, simple, easy drawing of a neuron. I would highly recommend drawing this in your notes. I just think it's helpful to have a pictorial representation. I think it helps us with memorizing the parts and functions and kind of understanding the overall way in which neurons communicate with one another. So if we look at the neuron, it travels from this direction to this direction, meaning the chemical messengers of neurons, those neurotransmitters, come in one side of a neuron, go through the neuron, and they exit the other side. It is a one-way street. You don't go the opposite direction. It's a one-way street. So what happens is those chemical messengers, those neurotransmitters, enter into these branches, which are called dendrites. And each of these can receive messages from other neurons. All of these are called dendrites. So that neurotransmitter is received by the dendrites. It enters into what we call the cell body or the life support system of the neuron. So it's called the soma or the cell body. That is kind of what keeps that individual neuron alive. And then that neurotransmitter travels down the axon that's surrounded by something called a myelin sheath that helps to insulate it, protect it, and allow it to move faster. And we'll talk a little bit more about the role of the myelin sheath in the axon in further slides today. But just so you are just getting the anatomy and structures down, the axon is this long area, and then this kind of yellow, this surrounding area is the myelin sheath that insulates and protects it. And then those messengers, those neurotransmitters, exit out what we call our axon terminals, or it's also known as the terminal branches of the axon. So if we can get in our notes just a drawing and labeling it, that's going to put you in good shape. One last part that's nice and helpful to show if we have multiple neurons here interacting, we can see here's one. We've got those dendrites taking that information, go into that cell body, that life support system, travel down that axon that's surrounded by the myelin sheath, and then it exits out those axon terminals or terminal branches of the axon. And as you can see, there's a little tiny space right here between the sending neuron and the receiving neuron, or between the axon terminals of my sending neuron and the receiving dendrites of my receiving neuron. That tiny little space right there is called the synapse, or the synaptic gap. That is important. So we don't ever have neurons that are fully touching each other. Rather, they have tiny spaces in between them for almost those neurotransmitters to exit out one side and then be absorbed by multiple different dendrites. If each dendrite connected to an axon terminal, then it would just be one communication to the other, versus one neuron sends out those neurotransmitters and multiple dendrites from different neurons can receive that message. So that's the anatomy. That's the structure of a neuron. How does it fire, though? How does a neuron get the neurotransmitter from point A to point B? And that's what this slide is all about. How does a neuron fire? Particularly when a neuron fires, it's called an action potential, meaning all of a sudden that neurotransmitter has been sent out of a neuron, sent into that synaptic area to be absorbed by another neuron's dendrites. That's called an action potential, right? That neuron has fired. In order to fire, what we see is there are these positive and negative ions that are hanging out in this axon. And as those ions are moving through the axon, we need to get enough positive ions into that neuron to get over what we call the threshold to allow that neuron to have an action potential. So what we mean by that is there needs to be enough positive energy entered into that axon, enough to get over that threshold or over that hill for an action potential to occur, for an action potential or a firing of a neuron to happen. Once it fires, it fires. If it doesn't fire, it doesn't fire. That's that all or none. We are either going to get over the threshold and an action potential is going to happen, or we're not going to get over the threshold and an action potential is not going to happen. That's that all or none principle. A neuron can't halfway fire. A neuron is either going to fire or it's not going to fire. Since this neural communication depends on this electrical stimulation, there is also what we call a refractory period. So after an action potential has happened, after that neuron has fired, remember that neuron has gotten over the threshold of those positive ions to give that neuron enough energy to fire, it then needs what we call a refractory period or a refresh period, where it has to kind of get itself back together to allow it to then get the energy again to fire another time. So there has to be a pause. A neuron just can't keep firing and firing and firing and firing. Rather, it needs that refractory period, that time in which it can recharge and re-energize itself. Finally, with how a neuron fires is we talk about neurons as being depolarized versus hyperpolarized neurons. Depolarized neurons are less likely to have an action potential. I'm sorry, I said that wrong. Depolarized are more likely to have an action potential. Depolarized neurons are more likely to have an action potential versus hyperpolarized neurons are less likely to have an action potential. So a depolarized neuron has more positive ions, leading it closer to an action potential. A hyperpolarized neuron doesn't have enough of those positive ions to get over that threshold and is less likely to have an action potential or for it to fire. Fires. Now we're going to talk about what are those little things inside of a neuron that are the little chemical messengers, and those are called neurotransmitters. So we are going to talk about different neurotransmitters, and each of the pictures on this slide represents one of those neurotransmitters. So the first one we're going to look up is acetylcholine. That is it is shortened to capital A, capital C, little h, in case you're interested, ACH, acetylcholine. I want you to look at the A plus in the top left corner of your screen. Acetylcholine helps us with muscle movement, learning, and memory. So I remember this by acing a test. You need to have a good memory to ace a test, or an ACE bandage put around for your sore muscles. We find that acetylcholine is also those neurons that produce acetylcholine have been linked to different disorders. For example, an undersupply of the neurons that produce this, so like the neurons that produce acetylcholine, when they deteriorate and start to die, we've seen that that is linked to Alzheimer's, which makes sense. If acetylcholine helps with memory, if those neurons that produce acetylcholine start to deteriorate, well, then of course your memory is going to be impacted. We also find that when we have an undersupply of acetylcholine, it also leads to a disorder called Myasthenia Gravis, which is muscle weakness due to kind of destruction of that neurotransmitter. So it is an autoimmune disorder. It's kind of a neuromuscular disorder. It causes weakness and slowness to our skeletal muscles. So our voluntary movements, such as walking, lifting, swallowing, even breathing, start to become more difficult. And so what's happening is our body is blocking, destroying, killing that acetylcholine, and that is slowing down and making our voluntary movements much more difficult. Our next one that we are going to talk about is dopamine. And you're going to look in the top right corner for this one where it says, why do you think they call it dope? And so dopamine it helps to coordinate physical movements. It is released in response to pleasurable experiences. So it improves our motivation. You might have heard about like a dopamine hit. They talk about when you go on your social media, you get this dopamine hit, that good feeling, that response, that release that happens when there's been pleasurable experiences. It also is linked to two particular disorders, one being schizophrenia and one being Parkinson's. And so I think about helping us remember that it's linked to schizophrenia and Parkinson's by thinking about dope. And when you take dope or drugs, maybe when you take too much of it, you may start to hallucinate or see things like happens with individuals who get hallucinations with schizophrenia. And in contrast, when you are going through withdrawal of a drug, so dope drugs, you get a withdrawal, you start to sometimes have the shakes. And in Parkinson's, that is one of the outcomes that we see as an individual has uncontrollable shaking of their muscles. So dopamine is released in response to those pleasurable experiences. It helps to coordinate those physical movements, and that oversupply is linked to schizophrenia, and that undersupply is linked to the muscle shakes in schizophrenia. The next one we're going to look at is right below that, serotonin. Serotonin is a mood regulator. It's a neurotransmitter that's tied to mood regulation and most closely associated with depression. An undersupply of serotonin has an association with individuals experiencing depression. So a lot of antidepressant medications are going to give you that serotonin in order to boost that mood. Norepinephrine is the next one. You're going to find the pin, which is underneath the A+. We're pulling the word pin out of norepinephrine. And think about if I were to poke you with a pin, you would probably have a hard time going to sleep. You'd be very alert, very aroused. You would be be responding and kind of enter into that fight or flight mode. Norepinephrine, it's released in response to stress, and it helps to initiate that fight or flight. It helps to help us to be more alert and aroused. We also see an association with norepinephrine linked to depression and undersupply. That's where, one of the diagnostic criteria or symptoms of depression is an individual might sleep too much or not sleep enough. An individual with an undersupply of norepinephrine might not be as alert and aroused and sleep often. And so that can lead to that diagnostic criteria, that symptom that we see in depression. But norepinephrine, when working correctly, helps us to be alert and aroused. And the next one is GABA. I want you to find the Yo GABA GABA on the screen. This neurotransmitter is what we call an inhibitory neurotransmitter. And what an inhibitory neurotransmitter is it's when you have an inhibitory neurotransmitter, it makes a neuron less likely to fire. So it's going to reduce brain activity. So this neurotransmitter, when it is released, actually reduces the chance of a neuron continuing to fire. And so what we find here is that an undersupply of GABA is linked to seizures and tremors, because what's happening is that our neurons are overfiring. And when there is too much firing that's happening in the brain, that can cause us to have seizures. Those too much electrical current happening is not a good thing in the brain. So that's where we get seizures or tremors happening. So if we don't have this GABA neurotransmitter, that is that inhibitory neurotransmitter that's reducing how much firing is happening of those neurotransmitters, that's where we see that uptick in seizures and tremors in an individual. The next one is glutamate. And for the picture for glutamate, it says, good day, mate. And this is the opposite. This is an excitatory neurotransmitter. So when this neurotransmitter is released, it increases the likelihood of a neuron firing. It increases kind of that brain activity involved in learning and memory. So if we have an oversupply of this, it can kind of cause like an overstimulation of the brain. They've found that there's a connection between the migraines that we receive, that if we have too much of this glutamate, we've got too much firing that's happening, it can cause headaches. So they've noticed a correlation between those two things. The next one is endorphins. That is end the pain with endorphins. So that's the big end, that big button with the word end on it. Endorphins regulate the perception of the pain. It's kind of like your brain's natural opiate. It enhances mood. It helps us to feel euphoric and pleasure. Oversupply of, if we take like opiate drugs that kind of suppress the natural body supply, can actually decrease how much endorphins we make on our own. So those endorphins are released and they allow us to regulate that perception of whether pain is happening or not. It enhances that mood. And so when we have those endorphins, it kind of can then lessen that perception a little bit of the pain. Substance P is the next one, and that's the big P that's on fire. This is also involved in perception of pain. This helps to transmit pain signals from the body to the central nervous system. So basically in the body, it helps to transmit those pain signals. So it helps to promote inflammation in response to an injury. So if we twist our ankle and then all of a sudden the ankle starts to swell, that's actually done to help our body, because if we can have that swelling, we're helping to promote that healing and staying off of it. And that inflammation is one of the keys that our body is working to fight whatever we did, like twisting our ankle. Individuals who have an oversupply of this could have chronic pain because they are constantly feeling the perception of pain. That pain messenger is constantly firing for them. Whereas it's good to have substance P. It's good that when we hurt our ankle, we get a signal that, hey, this hurts, that we get that inflammation. But if we get too much of that signal, we are going to be able to feel a lot of chronic pain that we don't want to have to feel. So those are the neurotransmitters. We are almost done, my friends. Stay with me. We have just one more slide left to go. Last slide to go is how to get more or less of a neurotransmitter. So let's say our body oversupplies or undersupplies something. Is there something that we can do about it? So you think about serotonin. If we don't produce enough serotonin, is it possible to get more serotonin in order to maybe not have depression? And the answer to that is yes. And that's by taking we have to look at kind of the options that we have for medications. So there's medications that work as an agonist, medications that work as an antagonist, and medications that work as reuptake inhibitors. So if we look at the difference between an agonist and an antagonist, agonists mimic, meaning if I take a medication that's an agonist, it will mimic the neurotransmitter that I have a decreased amount in. So it's essentially going to it's just going to produce synthetic or fake of that neurotransmitter to increase how much of it I have. Versus antagonists, this is given when we have too much, right? We've got too much of that neurotransmitter. We're going to take an antagonist, and what that's going to do is it's going to bind on those receptor cells. It's going to bind on those receiving dendrites so that when that neurotransmitter is sent out of the axon terminal into that synapse, it can't enter into the next neuron's dendrite, therefore reducing how much of that neurotransmitter is transferred through our body. The last way we can do it is with something called a reuptake inhibitor. And you may have heard of an SSRI before, or a selective serotonin reuptake inhibitor. But what this does is, if we go back to when we were talking about the synapse, right? We know that synapse is that gap between the sending and the receiving neuron. If we go back to that, what happens is that when a neuron fires, when a neuron has an action potential, those neurotransmitters are sent into that synaptic gap. Those dendrites are like hungry hippos, and they are eating and grabbing at all those neurotransmitters. Well, the neurotransmitters that are left in that synapse after the neurotransmitters have kind of all taken what they wanted, those neurotransmitters sitting in the synapse, a process called reuptake occurs, meaning the sending neuron reabsorbs and holds on to that neurotransmitter until the next action potential occurs, and it sends those neurotransmitters back out into the synapse. Hungry hippos, dendrites are grabbing at them. And then again, the reuptake occurs where they're sucked back up into those sending neurons, into those axon terminals. They're held there. Next action potential, they're sent back out. So there are medications that are called reuptake inhibitors, meaning they are medications that are going to block the reuptake from occurring, therefore giving you more of that neurotransmitter. So if that neuron has an action potential, sends out all the serotonin into the synapse, and then those dendrites, those hungry hippos, are grabbing at the serotonin, what normally happens is the rest of the serotonin that's in the synapse is reabsorbed into the axon terminal. But instead, the SSRI are going to block that from happening, so those serotonins that are in the synapse can't get back in. Therefore, they're forced to go into the dendrites, forced to send more of your neurotransmitter serotonin into your body. So, agonists, mimic, give you more. SSRIs, or reuptake inhibitors, give you more by blocking the reabsorption to force them to go back into the synapse and force them to go back into the dendrites. And antagonist, they block. They stop the dendrite from even receiving the neurotransmitter to begin with. So they bind to those dendrites and make it so those neurotransmitters can't even get into those neurons. So I know this was a long video lecture today. Lots of stuff. I promise we're going to do practice with this. I look forward to seeing you in class.